Welcome back. Let's go ahead and cover Unit 6, which is respiratory cultures and flora associated with respiratory cultures and pathogens. So, a respiratory tract infection happens when an organism comes in contact with the mucoso-epithelium um, lining and begins to multiply. So, if somebody comes up to you and coughs on you, you breathe it in, and that organism sticks to the mucosal epithelial cells, then all of a sudden you can become sick. The organisms may be colonizing normal flora or invading pathogens. You can be a carrier of strep A, for example, if you're colonized, or strep A can be a pathogen for you. And then colonization basically means organisms multiply and grow under optimal conditions without causing harm to the host. When I think of colonization, I think of organisms that live in the urinary tract, especially in elderly people that have indwelling catheters. Those organisms live there because it's dark and moist and warm, but they don't actually cause any problems for the host. They're just colonizing organisms. Let's take a look at the anatomy of the respiratory tract. In the upper respiratory tract, we have the nose, the throat, the oropharynx, which is basically the back of your throat, and the nasopharynx, which is the back of your nose. Then we have the lower respiratory tract, which includes the larynx, the trachea, the bronchi, the bronchioles, and the alveolar air sacs of the lungs. Now, normally there are a number of ways that our respiratory tract is protected from infection. For example, we have nasal hair, and nasal hair actually filters out and traps the organisms before they become pathogens. We have lysozyme, which is a chemical that actually lyses the cell walls of gram-positive organisms. We have antibodies, including antibody IgA or secretory IgA antibody, which is the antibody that coats the mucosal linings. Then we also have ciliated epithelial cells. They trap things that we breathe in and we can cough those out. We have macrophages and our cellular defense mechanisms. We've got macrophages, neutrophils, and monocytes, and those are all part of our second line of defense, which is phagocytosis and inflammation. We have the ability to cough, sneeze, and swallow, either coughing out organisms that could be pathogens, sneezing them out, or swallowing them down and expelling them that way. And then finally, we have normal respiratory flora. And these are organisms that live in the upper respiratory tract that compete with nutrients and things of that nature with pathogens. I want to start today by talking about an organism that can be a pathogen of the respiratory tract, and this organism is called Haemophilus species. This is an organism that is small. In fact, we call it a tiny gram-negative rod, and it's non-modal. In fact, sometimes on gram stain, it appears as a very small cocci, but they could be pleomorphic also, where you see a mixture of rods and cocci all in a pure culture. They're catalase and oxidase positive and will reduce nitrates to nitrites. They're fastidious and they do require chocolate agar to grow on, at least Haemophilus influenza does, and they do require an environment rich in carbon dioxide. They will not survive refrigeration, and they require blood factors for growth, and those blood factors would be hemin and NAD, which are also known as factor X and factor V. Some species are normal flora, and other species, like Haemophilus influenza, can be pathogens. So about 10% of the population does harbor Haemophilus species in the upper respiratory tract. Now, Haemophilus influenza may be normal flora, or it could be a pathogen, and a lot of times it is a pathogen. In order to get it to grow on micromedia, we have to provide both the X and the V factor for growth. The X factor is hemin and the V factor is NAD. Chocolate agar is an ideal media for Haemophilus influenza because of the lysed red cells in the media. It provides both the X and V factors. It's not going to grow on blood agar unless there's a source of the V factor. Blood agar only provides the X factor, but if Staph aureus is growing there, it will provide the V factor, and what you'll find is Haemophilus influenza will actually satellite around the colonies of Staphylococcus aureus. So here is an example and an image of satellitism, where we have Staphylococcus aureus, which are the large yellowish colonies on the media, and they're providing the V factor. The X factor is already in the media. So colonies of Haemophilus influenza will actually satellite around the Staphylococcus aureus, and they appear like little gray dots. So there's many types of Haemophilus species. Haemophilus influenza primarily being the one that we care about because it can be a pathogen. But we can differentiate Haemophilus species by exposing it to the X and V factors. So we also want to put the Haemophilus species on horse blood agar. And some Haemophilus species will be beta hemolytic on horse blood agar, but Haemophilus influenza is actually gamma hemolytic on horse blood agar. And it fails to grow on sheep's blood agar unless Staph aureus is present. So here's an example of Haemophilus influenza that requires both the X and V factor. It's on a type of media known as brain heart infusion agar, and you can see that dust growing around the X and V factor is Haemophilus influenza, and you can also see that Haemophilus influenza is the dust that's growing between the X and V factors, and that's because it does require both. So like I've expressed, Haemophilus influenza can be normal flora, but many times it is a pathogen. It can cause infections of the pharynx. It can cause infections of the epiglottis, which is swelling of the epiglottis. The epiglottis is the flap of skin that divides your respiratory tract from your gastrointestinal tract in the back of your throat. Haemophilus influenza can cause sinusitis. It can cause eye and ear infections and bronchitis and pneumonia, and often that is a secondary infection from a viral infection primarily. Haemophilus influenza can cause endocarditis, cellulitis, and meningitis. There is a vaccine that has reduced Haemophilus influenza meningitis cases. The vaccine is made with toxoids and or fractionated materia. Type B Haemophilus influenza is the most pathogenic strain of Haemophilus influenza, leading to the vaccine development. And there are about 16,000 cases of Haemophilus influenza in children every year. The second pathogen of the respiratory tract that I want to talk about is Streptococcus pyogenes, which is also known as Streptococcus group A. Unfortunately, we're probably all too familiar with strep throat, and that is primarily the disease that Streptococcus pyogenes causes. There is a correct way to collect this patient sample. And by doing that, we want our patient to sit comfortably and to tilt their head back at about a 45-degree angle. We're going to take a sterile tongue depressor and press the tongue down with the tongue blade. And the patient should say ah as they're opening their mouth here. And what that does is it prevents the patient from having a gag reflex. And I will tell you, patients that have strep throat, they often are pretty gaggy. They usually are very, very sick and not feeling well at all. Then what we'll do is we'll take a sterile swab and we're going to swab the tonsillar areas on both the left and right hand sides, and also behind the pharynx, which is behind the uvula, which is that little dangly thing in the back of the throat. We want to remove the swab without touching the inner cheeks, teeth, or tongue, and the reason why is we don't want to pick up all that interfering normal flora. Instead, we only want to culture out the Streptococcus pyogenes as best as we can. There's a couple of correct ways to culture Streptococcus pyogenes, also known as Strep A. We can take a blood agar and we can actually stab it with the loops, and what we're doing here is pushing the organism down into the anaerobic zone of the plate. Or we can incubate that plate anaerobically and use a 4-streak method for isolation. Both of those techniques are acceptable for isolating Streptococcus group A. We never want to use a capnophilic environment. Then we can put a couple discs onto our plate in order to screen for Streptococcus group A. The SXT disc is a disc that we use to differentiate between Strep A and Strep C. Strep A is going to be resistant to the SXT disc, while Strep C is going to be sensitive to it. And by the way, Streptococcus group C can cause a sore throat. It just doesn't lead to all of the nasty complications that Strep A does. The bacitracin disc is the A disc, and that helps us eliminate normal flora and helps us confirm strep A. It's called the A disc. The bacitracin disc is called the A disc because Streptococcus pyogenes is going to be sensitive to the bacitracin disc. Then metabolically, we can identify our Streptococcus pyogenes by studying the PYR test. It's going to be positive. The esculin test will be negative. And then the best way to identify Streptococcus group A is by serologically typing. And what we're doing is we're taking latex particles and actually linking those latex particles up with the antigen ripped off the organism Streptococcus. And if they link up together, you will get agglutination that you can visually see. And those agglutinins, if they're present, we're gonna report that out as positive for strep A. So here's a really nice picture of what Streptococcus pyogenes looks like on sheep's blood agar. They are small colonies at 24 hours. They are dry and they have large zones of beta hemolysis. Just to remind you, beta hemolysis is when the organism produces an enzyme or a toxin that completely lyses the sheep's blood cells in the media. And you can see there that there is a clear zone around the colony, and that is called beta hemolysis. Strep throat or streptococcal group A pharyngitis is the most common infection of the upper respiratory tract. The organism will colonize in the pharynx and multiply and produce its toxins there. The back of the throat above the epiglottis is called the oropharynx. What you're looking for there is white spots on the tonsils, a grayish furry tongue, a swollen uvula, and even reddish tonsils as well. The incubation period is about 2 to 4 days from the time of contact with someone that has strep throat. The symptoms are fever, malaise, headaches, chills, sore throat, painful swallowing, edema, which is swelling, and then exudate covering the tonsils and swollen lymph nodes. Pharyngitis is most of the time is about 60% from viruses, and 40% of the time pharyngitis comes from a bacterial source. The reason Streptococcus group A is so important is because it can lead to other things such as sinus infections, middle ear infections, infections of the mouth, or it can lead to rheumatic fever, which is an infection of the heart valves, or bacterial glomerulonephritis, which is an infection of the nephron of the kidney. In acute rheumatic fever, the toxins of Streptococcus group A affect the heart valves. In acute rheumatic fever, the patient has a fever, increased erythrocyte sedimentation rate, and that's basically a lab test that determines how fast your red cells fall, inflammation of the heart muscle and valve damage, joint pain, and twitching of the facial muscles. In acute glomerulonephritis, this is when immune complexes form in the kidney and actually destroy the nephrons of the kidney. Symptoms of that would be edema around the eyes and ankles, reduced urine output, a smoky red colored urine. Urine samples are going to be positive for blood and protein, and you're also going to see red cells and red cell casts microscopically, and the white cells are also going to be noted in the urine. There is a lot of normal flora that lives in the upper respiratory tract. The growth of these organisms occurs when samples are collected up through the mouth. So for example, a sputum sample that is coughed up and collected through the mouth is going to grow quite a bit of commensal flora. But some of the commensal flora of the upper respiratory tract would be miscellaneous alpha and gamma streptococcus, not necessarily Streptococcus pneumoniae, but other alpha hemolytic streptococcus. Some of our beta hemolytic streps, not strep A, C, or G, those can all cause a sore throat. Coag negative staphylococcus, diphtheroids, which are the gram-positive rods, also known as Corynebacterium, and then Haemophilus parainfluenza, and finally some of your Neisseria species, not gonorrhea, not meningitis, but other Neisseria species such as Neisseria lactamica and things like that, Neisseria sicca. There can be a colony or two of enterics or non-fermenters in the upper respiratory tract, and that's due to reflux. Most of the enterics and non-fermenters live in the gut, and so they should actually stay in the gut and not actually come up into the respiratory tract. A colony or two of Staph aureus, Strep pneumo, Moraxella catarrhalis, and Haemophilus influenza, and even yeast can be normal if there's a bunch of normal flora. If there's a lot of Staph aureus or a lot of Strep pneumo, etc., then that's going to be a problem. Yeast in the respiratory tract may or may not be a pathogen. Could it be an opportunist? Could it be colonization? We'll leave that for the physician to decide. But usually, if you see yeast in the respiratory tract, you do want to identify it and let the doctor know that it's there. It can be a pathogen most commonly in people that are mechanically ventilated, however. So here's kind of a list of most of our respiratory pathogens, and these are the lower respiratory tract pathogens. Miscellaneous viruses, of course, like influenza A, parainfluenza, and things of that nature. Bordetella pertussis, that's the organism that causes whooping cough. Corynebacterium diphtheriae causes diphtheria. Legionella, Mycobacterium species, that is what leads to tuberculosis. Streptococcus group A, that causes strep throat. Enterics and non-fermenters, and Mycoplasma pneumoniae, that's usually found in small children. Other organisms that can be pathogens if they're there in great numbers would be Staphylococcus aureus, Streptococcus pneumoniae, Moraxella catarrhalis, and Haemophilus influenza, and maybe yeast. Let's take a look at bacterial versus viral pneumonia and kind of what the symptoms and onset kind of look like. So in a case of bacterial pneumonia, the onset is sort of sudden with a bang, and in a viral pneumonia, it comes on kind of gradually. You have one big single kind of intense chill with the bacterial pneumonia, and you always may feel a little bit chilly when you have viral pneumonia. You're acutely ill from the onset with bacterial pneumonia, and you may appear well with a slight cough or fever initially with the viral pneumonia. Your cough with the bacterial pneumonia is productive and purulent and bloody, so you're coughing up a bunch of stuff, and usually with viral pneumonia, it's not productive at all. You can have a temperature with both types of pneumonia, but usually it's higher with bacterial pneumonia. You can develop pleurisy, but again, that's more common in bacterial pneumonia than viral. And your white cell count usually goes up in a bacterial case of pneumonia as opposed to a viral pneumonia. Bordetella pertussis, often called whooping cough, named after the whoop sound that people make trying to catch their breath after prolonged coughing. There are over 350,000 deaths per year, and those virulence factors of Bordetella pertussis are membrane proteins that help it attach to the host, and the pertussis toxin, which actually interferes with the host proteins, Humans are the only host, and the symptoms are fever, vomiting, cyanosis, and cough. To collect this, we would take a nasopharyngeal swab and collect it at the bedside. Usually a nurse or some other medical personnel, a doctor or a nurse, will collect this at the bedside, and they'll put it into a slant known as the Reagan-Low Deep, and then it'll be brought to the laboratory. And then in the laboratory, we will take that swab out of the Reagan-Low media and put it on a media with charcoal agar within the next couple days, and we'll look for things that look like mercury drops on the media. Corynebacterium diphtheriae causes diphtheria, and it's not common in the United States because we get a vaccine for it. Usually we get a vaccine between 6 weeks old and 12 months of age. It is common in developing countries such as Europe and South America. The disease produces a low-grade fever, pharyngitis, and a blocked airway. It's actually quite a bit worse in symptoms than Streptococcus group A because it causes the tissue around the throat to actually die, and that's called necrotic throat tissue death. Okay, like I said, there is a vaccine available. It is virulent because of the diphtheria toxin. It's transmitted from aerosols, And the media of choice is cysteine tellurite blood agar or modified Tinsdale agar. And it's going to be brown or black with a brown halo on modified Tinsdale agar. And you can see that in the bottom left image. It does require 35 degrees incubation to grow it, and it is a gram-positive rod. So out of all of the Corynebacterium, which are called the diphtheroids, this is probably the only one that's a pathogen most of the time, and it's called Corynebacterium diphtheriae. A little bit more, just to kind of review Corynebacterium diphtheriae. It's an aerobic gram-positive rod without spores. It's non-motile. It appears as Chinese letters or picket fences, or they're palisading on gram stain. They cause diphtheria or diphtheria pharyngitis due to the diphtheria exotoxin. Symptoms would be very hard time swallowing, fever, cardiac and nervous system issues. And like I mentioned, there is a vaccine available, the DTaP, which is the diphtheria, tetanus, and pertussis vaccine. And you can culture it on serum tellurite or Loeffler's media. The colonies are going to be gray or black. Mycoplasma pneumoniae is an organism that is considered an opportunistic respiratory pathogen. It causes somewhere between 10 and 30% of all pneumonia cases. It's very difficult to culture, and it might be best to diagnose it serologically, where we're looking for IgM or IgG antibody levels. It can be treated with tetracycline or erythromycin. And if we do want to try to culture it, we can culture it on a media known as PPLO media. And this is called pleuropneumonic-like organism media. And that's because it was first discovered that mycoplasma was in cows that have pleural pneumonia. It is what we call a soft skin organism, and it's one of the smallest free living organisms at, it's very, very small, at about 0.05 microns. It's not going to gram stain because it has weak, thin cell walls, but if you were to try what's known as a Dean stain, you would note the fried egg appearance that you can see on the right there. Legionnaire's disease accounts for about 2 to 15% of community-acquired pneumonias. About 8,000 to 18,000 hospitalizations occur annually because of Legionella or Legionella pneumophilia. It causes Legionnaire's disease and Pontiac fever. Technically, it's gram-negative, but it doesn't stain easily with the gram stain. Giemsa and Diff-Quick stains work well. It is fastidious and requires L-cysteine and iron salts for growth. The media of choice to grow this organism is BCYE media, which is charcoal yeast extract agar, and it's going to appear as a ground glass appearance, and you can kind of see that in the image on the right. It's kind of got that sparkly sheen to it. It is slow to culture, and the best way to diagnose it would be with a direct fluorescent antibody test, and there's a nice urine antigen test as well. It's called the Binax urine antigen test, and it's 97% sensitive for the Legionella antigen, but we do have to confirm it usually with culture. Now, Legionella is associated with a couple diseases. One is called Legionnaire's disease, and one is called Pontiac fever. Pontiac fever is a lot less severe than the Legionnaire's disease, and when you have Pontiac fever, you kind of feel like you have influenza. When you have Legionnaire's disease, you have a 15 to 30% chance of dying. You have fever, chills, chest pain, cough. You're making a lot of sputum and diarrhea, so the signs and symptoms kind of run the gamut. It can develop into pneumonia with lung abscesses, and then that can turn into renal and liver failure. You can have delirium and disorientation as well. Not a fun disease to have. Pontiac fever, on the other hand, isn't deadly. You have flu-like symptoms. Usually you don't develop into pneumonia and you don't develop any lung abscesses. There's no renal or liver failure, no gastrointestinal involvement, and no nervous system involvement either. So it's quite a bit less severe than Legionnaire's disease. Moraxella catarrhalis is a normal organism in the upper respiratory tract, although it can be an opportunistic pathogen. It can cause things like pneumonia, and usually I've seen it cause sinusitis. I think it's the #1 cause of sinusitis, actually. It can cause otitis media, which is a middle ear infection. It can cause blood infections and also systemic diseases. It grows fine on blood agar and chocolate agar, and it's aerobic. It doesn't require 3 to 10% CO2. There's a number of ways you can identify Moraxella catarrhalis. The first two steps you want to do is a catalase and an oxidase test. It's going to be oxidase positive and catalase positive. And then more so than anything else, you want to see if it has the butyrate esterase enzyme. And if it does, it's going to be positive for that. And that's a confirmatory test for Moraxella catarrhalis. Now, we talked about the catalase test when we talked about Staphylococcus way back in Unit 3. Staphylococcus are vigorously positive for the catalase test, where they bubble with a 3% H2O2 solution. Now, Neisseриas and Moraxellas, on the other hand, they may require a 30% solution of H2O2 in order to get accurate results. You can see the image on the left is a negative catalase test, and the image on the bottom left is a positive catalase test where you have vigorous effervescence when mixing the solution with the organism. I've mentioned the cytochrome oxidase test before, but it's been several units back. So let's just go ahead and refresh our memory. Neisseria species and Moraxella are cytochrome oxidase positive. And it's where we have the enzyme cytochrome oxidase in the presence of oxygen is going to form a colored compound known as indophenol when combined with 1% tetramethyl p-phenylenediamine. So your organisms like staph and streps are negative for oxidase, and your organisms like Neisserias, Moraxellas, and non-fermenters are going to be positive for the oxidase enzyme. And there's many methods available for this test, including what's known as the swab method, where you take the organism and put it on a swab and then add your reagent to the swab and see if it turns purple. Then we've got the filter paper method, where you add the reagent to the filter paper and then you smash your bug into the filter paper. If it turns purple, it's positive. And then there's the plate method, where you add the reagent directly to the organism on the plate. So there's several methods available for this test. The butyrate esterase test is the confirmatory test for Moraxella catarrhalis. This is also known as the catarrhalis test disc method. And basically what we're doing is we're taking a dried paper disc that's impregnated with indoxyl butyrate, and we're smashing our organism, our unknown organism, onto that disc. We're going to set a timer for 2 minutes, and at the end of 2 minutes, if our disc turns blue or green, it indicates a positive test for the butyrate esterase enzyme. All right, this brings us to an ominous organism known as Bacillus anthracis. Commonly, well, not commonly, thank goodness, but it is the cause of anthrax. And it's actually been used in biowarfare in Japan, the former Soviet Union, United Kingdom, and Iraq. And in 1979, the Soviets accidentally released some during weapons testing and killed 66 people doing the research. It is an aerobic gram-positive rod. Other species of bacillus are environmental commensal organisms, but Bacillus anthracis is a pathogen. It's encapsulated in a spore former. It's catalase positive and non-motile. Other bacillus species are motile. And there's 3 forms of anthrax, including cutaneous, inhalation, and gastrointestinal. The diagnosis for Bacillus anthracis is gram staining, culturing, PCR, polymerase chain reaction or molecular identification, or fluorescent techniques. Mammals coming in contact with spores by consumption of contaminated food, skin contact with spores, or inhalation of aerosols is what leads to anthrax. Okay. There is a vaccine called Biothrax. It's given to military personnel. And there's a few virulence factors. One is the fact that it's encapsulated with the LT toxin, which is the lethal toxin. And usually this is not a problem for most people in the United States. However, it is an occupational hazard when working with herbivore animals, such as people that are veterinarians, people that are ranchers, and things like that. So here's a gram stain of Bacillus anthracis. The gram stains are on the left. You can see they're a large gram-positive rod with a spore. And then the bottom left image is where you can see the large gram-positive rods as well. And then on the right-hand side, you can see what's known as the medusa head colony morphology. And on blood auger, they do have kind of this trailing paisley type of tail, or it looks like the head of Medusa, where you have the head and then you have the hair, and it looks kind of like snakes coming out from the hair. And then, of course, the best thing about talking about anthrax is all the gory pictures that I can include. So these are pictures of eschers that are formed from cutaneous anthrax. And again, this is usually people that are working in the veterinary or ranching industry that come in contact with animals that have anthrax. It looks pretty painful, but it's actually the outside edge that is painful. That inner black area of the escher is not that painful. And then in the image in the bottom left, that shows an image of pneumonia caused from Bacillus anthracis. When I think of Mycobacterium species, most often I think of the organism tuberculosis. This is one of the Mycobacterium. It is a major global public health concern. There are about 8 million cases of tuberculosis every year, according to the World Health Organization, and about 3 million deaths per year. Many factors account for the increased incidences in the U.S. In the U.S., there's about 80,000 cases per decade. But some of the reasons why there are factors that have increased the incidences of tuberculosis can be organism drug resistance, the AIDS epidemic, and the healthcare infrastructure collapse. Many species can cause disease. It's not just Mycobacterium tuberculosis that cause disease. And they're not easily gram stained. They're actually termed gram invisible because they have high cell wall lipids and waxy acids in their cell walls. They are called acid-fast bacillus, and that's because they are not easily decolorized with acid alcohol, thus they're called acid-fast. They're slightly curved, bent, or branched bacillus, aerobic with no spores, and they may demonstrate what's known as serpentine cording, and that's where you have bacillus that chain and form cords. Acid-fast organisms don't stain very well with gram stain. They're actually called gram invisible, and they will stain as ghost forms or beaded positive rods. There are a couple stains that we can use to identify these organisms on a direct exam. We can use the Kenyon's carbofusion stain or the auramine rhodamine stain. The Kenyon's carbofusion stain is considered a cold stain and takes about 5 minutes. The reason it's called a cold stain is because there's no heating required like with the Zeal-Nielsen acid-fast stain, which does require heating of the reagents. The auramine rhodamine stain is a stain that takes about 20 to 30 minutes or so. It is a fluorescent stain and it's much more sensitive than the Kenyon's carbofusion stain. A minimum of 300 oil immersion fields should be examined. With the Kenyon's carbofusion stain, the organisms look red or pinkish on a blue background, and with the auramine rhodamine stain using a fluorescent microscope, the organisms appear fluorescent orange on a black background. According to CLSI, the Clinical Lab Standards Institute, stains have to be reported within 24 hours of order by the doctor. Mycobacterium typically are pretty slow-growing organisms. There are Mycobacterium fortunum Chelonia abscessus that take about 3 days, but most of your Mycobacterium organisms that cause disease take about 2 to 3 weeks to grow. And so we want to culture them to medias that don't dry out and medias that we can recognize their tiny colonies very quickly. So we use egg-based media, which is called Lowenstein-Jensen. It contains eggs, egg yolks, potato flakes, salts, glycerol, and malachite green. And basically, this type of media has a longer shelf life and doesn't dry out very quickly, so we can culture those slow-growing organisms. Malachite green actually inhibits the growth of normal flora. We want to inhibit normal flora from interfering with our isolation of Mycobacterium species. An agar-based media that we would use is called Middlebrook 7H11. The media itself is transparent, and we can see the microscopic colonies underneath a microscope when they first start to grow, and so it's a nice media for rapidly identifying slow growers like Mycobacterium. Mycobacterium can grow in broth media a little bit quicker, a little bit more rapidly than they grow on solid media. And one type of broth media that we can use to culture Mycobacterium is called Middlebrook 7H11. There's also another type of broth media called midget broths, Mycobacterium growth indicator tubes, that consists of 7H11 media and bovine albumin and oleic acid. We can use those medias to grow our organisms a little quicker. So here are some pictures of Mycobacterium tuberculosis. In the upper left-hand image, you can see the serpentine cording of the organisms growing on 7H11 media. And that would be what you see under a 10x objective when you put your plate on the microscope stage. On the right, you can see a Middlebrook 7H11 media with some colonies of Mycobacterium tuberculosis that are pretty old, actually. They are called buff and rough, and they look kind of buff in color and kind of rough or bumpy. And then a close-up of Mycobacterium tuberculosis is on the bottom left image. There are a number of specific populations that are at risk of developing Mycobacterium tuberculosis. Some of those populations include ethnic minorities, homeless people, prison inmates, alcoholics, and IV drug users, and also the elderly. The Mycobacterium tuberculosis organism is carried in airborne particles. It's carried and transmissible by coughing patients. One way we can test people to see if they've been exposed to the antigen would be the purified protein derivative test or the quantiferon test. The purified protein derivative test is when they take the antibody and they inject it under your skin, and if a wheal forms that gets large, it usually means that the patient has been exposed to the antigen. The quantiferon test is a very specific test that measures interferon gamma, a substance that is released by white cells if Mycobacterium tuberculosis antigen is present. And again, that's a blood test. Symptoms of Mycobacterium tuberculosis would be things like cough, weight loss, low-grade fever, difficulty breathing, and chest pain. Mycobacterium tuberculosis is actually a complex of organisms. It includes Mycobacterium africanum, and this is an organism that is seen in countries like Africa that is more likely to cause tuberculosis-like illness. They are non-chromogenic, which means that they basically don't make any pigments in the light or in the dark. They stay rough and buff, and they grow fine on Lowenstein-Jensen or Middlebrook 7H11 at body temperature, which is 35 to 37 degrees. They note serpentine cording, and they will accumulate niacin, and they will also be positive for nitrate reduction, and they are catalase negative. The reason I mention those things is most other species of Mycobacterium are niacin negative, nitrate negative, and catalase positive. If you wanted to treat a person with Mycobacterium tuberculosis, you could do so with certain antibiotics. They usually give 2 antibiotics at a time to treat this organism, and you can be on antibiotics for up to 18 months. Some of those antibiotics would be rifampin, ciprofloxacin, and isoniazid. But again, these treatments are very rough on the liver, and so our patients would be constantly checked for their liver enzyme activity. I want to switch gears now and talk a little bit about eye cultures. Eye cultures can be considered respiratory cultures, or they can be considered wound cultures, but there are pathogens of the eyes that are pathogens we've already talked about, including chlamydia trachomatis, and that can go from mom to baby. If mom has chlamydia and the baby goes through the birth canal, the baby can actually pick up chlamydia trachomatis. Haemophilus influenza, Neisseria gonorrhea, and Staphylococcus aureus. These are all pathogens of the eye. Chlamydia trachomatis is kind of an interesting organism. Sometimes they're considered a parasite, and other sources consider them an intracellular bacterium. There are about 900,000 new cases every year in the United States, and it is the most commonly isolated genital pathogen. It is the cause of many infections, and sometimes those infections are asymptomatic and easily transmissible. They are considered a sexually transmitted disease, and they can be transmitted from mom to baby. In females, this disease can lead to pelvic inflammatory disease. Specimens are obtained from cervical scrapings or mini-tip collection, and that's a mini-tip swab. And we can diagnose chlamydia trachomatis by using cell cultures. And again, that's when we are culturing to living cells. Giemsa stains, fluorescent antibody, or DNA probes. Okay. They won't stain with gram stain because they won't stain with gram stain, but they do have a cell wall that's similar to a gram-negative organism. Let's talk a little bit about Neisseria species. Neisseria organisms are gram-negative diplococci, and they are often shaped like kidney beans. They're catalase positive and oxidase positive, non-motile, non-spore forming, and many are normal flora, including normal flora of the upper respiratory tract. There are two main pathogens, Neisseria gonorrhea and Neisseria meningitidis. Neisseria meningitidis actually colonizes about 30% of the population in the nasopharynx without being a pathogen. Neisseria gonorrhea, on the other hand, can be a pathogen of the throat, but is more likely to be a pathogen of the genital tract. Like I said, Neisseria species can be normal flora of the respiratory tract, and that would include species like Neisseria sicca or Neisseria lactamica. The pathogens are Neisseria meningitidis and Neisseria gonorrhea. Neisseria meningitidis is often a pathogen of the upper respiratory tract and even in the central nervous system. If we wanted to identify a Neisseria species, we could do so by gram staining it. A gram stain would show gram-negative diplococci. They would be catalase positive and oxidase positive, and we would use carbohydrate utilization tests to identify certain species. And that would be, do they ferment things like lactose, sucrose, maltose, and dextrose? So here's an example of the organisms and what sugars they ferment. If you look at the tubes over there in the image on the right, you can see that they are maltose, lactose, dextrose, and sucrose. And if we put our organism into those tubes and then incubate them, Neisseria gonorrhea is going to ferment glucose, but maltose, sucrose, and lactose are all negative. You can remember that because Neisseria gonorrhea begins with a G and they are glucose positive. And glucose, by the way, is the same thing as dextrose. Dextrose is a sugar that originates outside of the body, and glucose is the same sugar, but it's inside the body. Neisseria meningitidis, on the other hand, are going to be glucose and maltose positive, sucrose and lactose negative. You can remember that Neisseria meningitidis is maltose positive because it begins with an M, maltose positive, meningitidis with an M, and they're also glucose positive. Neisseria gonorrhea can colonize the urethra, the cervix, the anus, the throat, and the conjunctiva. It will cause inflammation of the mucosa, and it's highly transmissible because it has pili, and the pili of the cells hang on to the mucosa of the host. The host can remain a carrier and a source of infection to others, and Neisseria gonorrhea can lead to infection of the fallopian tubes, the Bartholin glands, the periurethral abscesses, and arthritis and lesions. So just to summarize, the best way to identify an isolate of Neisseria gonorrhea is the microscopic morphology. It's going to be a gram-negative diplococci. What they look like on the macroscopic plate, they're going to be small, gray and translucent, and only on chocolate agar. They won't grow on blood agar. Carbohydrate utilization, they are glucose positive, and then you can use immunological or monoclonal antibodies to identify this disease in patients as well. Treatment for Neisseria gonorrhea would include treatment with penicillin, cephalosporins, and fluoroquinolones. It used to be that most strains of Neisseria gonorrhea were sensitive to penicillin, but now we are seeing that the third and fourth generation cephalosporins are usually used to treat Neisseria gonorrhea. And also the fluoroquinolones, but fluoroquinolones can be dangerous to people that are 18 years of age or younger, so we tend to see more physicians prescribing the third and fourth generation cephalosporins.